Good morning, biology students, and today we are going to be going over chapter 1 of the biology course. Now, you do have these readings as well in Canvas, but this is just kind of an additional resource for you. So this outline is going to cover the characteristics of life, evolution and the classification of life, process of science, and the science and challenges facing society. So first of all, the biology is the study of life. So that is what biology means. Bio for life, ology is the study of. So there's a lot of different types of groups of science. So you've got zoology is the study of animals, ecology is the study of living things interacting, meteorology is the study of weather, anything that ends in ology is the study of. So biology, this course is going to cover this all of living things. And keep in mind that this isn't just animals, this also includes plants. So there's great diversity among living things. Living things are composed of the same chemical elements as non-living things. So a desk or a chair has atoms, and we have atoms as well. The difference is just how they're combined together. So for instance, if I gave every person a set of Legos that were exactly identical, same colors, same sizes, everything's the same, and I told you to build a house, well, my house would look different from your house. And that's because really what makes something living versus non-living is how those individual Lego pieces are put together. And our body does have a blueprint for those Lego pieces, which is DNA, which we'll talk about at a later date. So they have to obey the same, whether we're living or non-living, the same physical laws and chemical laws that govern the universe. Despite diversity, all living things share the same basic characteristics, and so these characteristics we see as ability to reproduce, ability to adapt, and having DNA. These are all things that all living things must have in order to survive. Levels of organization. This is something that you'll probably be tested over, so make sure you're familiar with it, but it's starting at the most basic and building to the more advanced. So the idea here is that you have small things and you combine those small things to make bigger things, and you combine those to make even bigger things, and everything builds on the layer that's below it. Starting with atoms. Atoms are the smallest unit that is considered alive, or sorry, not considered alive. It's the smallest thing that makes up any component, living or non-living. The cell is the smallest thing that's considered alive or can be alive. So you do have single-celled creatures, or you can have multicellular creatures. But atoms make up everything, and then they combine together eventually to make a cell. Once you get to a cell, you can combine those cells together to make a multicellular organism, or not, because again, some living things are just one cell big. Each level of organization is more complex than the one that's before it. And as biological complexity increases, each level acquires new properties. This is called emergent properties. Emergent property means that you see something that you haven't seen before. And this is going to be happening as the more complex organisms evolve. So starting with an atom, that's our most basic unit, and then you can make a molecule. So a bunch of atoms together make a molecule. Once you have these molecules, you combine the molecules together, you can make a cell. Combine the cells together, you get a tissue. So this is a group of similar cells that are working together to perform a similar function. And then you have an organ. Organ is a group of tissues that's working together to perform a similar function. Then you've got organ system, organism, and by the time you get to organism, you can also get up to species, or sometimes these are called a population, which is a group of the same kind of organism that's living kind of in the same place. So if we're in a classroom, there's a population of students in that classroom. You also notice that this chart is really nice because it is taking into account the animals versus the plants, so it's showing them side by side. So here's our population, is the group of all the living things of that type in the same environment. And then we have a community. A community is all of the different kinds that are living there. So not just all the elephants, but all the elephants and the zebras. Ecosystem, you take into account all the living and the non-living things, so like the water and the grass, or sorry, the terrain, like if there's hills or if there's mountains and the rocks, those kinds of things. And then biosphere is the top of this organization. Sphere meaning circular, and the bio meaning life. So basically, it's looking at the earth as a whole. So the biosphere is the zone of air, land, and water where the organisms exist. And then if we're going back down the row, we've got ecosystems, community plus its physical environment, again like water and hills. Community is a collection of the same populations. Population is the same organism. And that species, what defines a species? It's a group of similar organisms that can breed and reproduce fertile offspring. So a species is similar enough that it can breed and its offspring could also breed. An organism is formed when the organs are joined together. Organ system is after that. So think of like as an organism like a person. An organ system would be the digestive system. An organ within that digestive system would be the stomach. Then you've got the tissues of the stomach, so the muscular tissue that lines the stomach, the cells that make up that muscular tissues, the molecules, and then the atoms. Now we can also talk about energy. Energy is the ability to do work. Energy is really important because it is what allows us to function. And then we've also got something called metabolism. This is how chemical energy is broken down within the cell. So our bodies use, and most living things use something called chemical energy. But there are other forms of energy out there, such as light energy, sound energy, water energy, those kinds of energies, air energy, but the type that we use is chemical energy. So we can't use those other energies. We need to change it into a usable form. So the way that our energy gets turned into a usable form is through photosynthesis. So obviously we're not doing photosynthesis, the plants are. And then we will eat the plants and use the energy that they have created for us. But we cannot harness light energy. We could go out and we could stand in the sun as much as we want. We get a sunburn and we get vitamin D, but we get very little else. But the plants, when they're out in the sun, they can harness that sun energy and turn it into a chemical energy that their cells can actually use. Well, we can take that from the plant and we can harness that chemical energy for ourselves as well. Now, ecosystems are characterized by chemical cycling and energy flow. So chemicals are not used when an organism dies. So think of, for instance, when we are carbon-based life forms. So carbon is something that stays within our bodies. And when we die, it just turns into, it gets recycled in the environment. So energy does not get recycled, but nutrients do. So I'm sure you've all heard of the water cycle before and how you've got evaporation and condensation and precipitation, and the water goes in a big circle. Well, nutrients do that as well. You've got carbon that goes from a living thing into decomposing back into the soil, and it can be turned into fossil fuels, and fossil fuels can be burned and release the carbon back into the atmosphere. So energy goes in one direction, but nutrients can be recycled. Chemicals move from producers to consumers to decomposers, and when we die, we decompose and our nutrients are released back into the soil. But energy does not do this. Energy can only go in one direction, and typically that's up the food chain. So the sun energy is taken by the plant and harnessed by photosynthesis, and that photosynthetic energy is then kept within the plant. So a plant, let's say, has 100% of the energy that's available. Well, as you move up the food chain and something eats the plant, say a rabbit eats the plant. Well, the rabbit is not going to get 100% of that energy that the plant has to offer. It can only get 10% because 90% of the energy is actually lost as heat. So when that energy is lost as heat, that means the rabbit only got 10%, which means it needs to eat even more plants in order to get the energy that it needs. Well, let's say a snake ate the rabbit. The snake can only get 10% of the energy from the rabbit. So what is that, like 1% that originally came from the plant? So it needs to eat more too. This also means that as you go up the food chain, more and more energy is lost as heat. And the animals that live at the top of the food chain, like the main carnivores, those are not going to have a lot of energy to offer. So this is also why big animals like the blue whale can live off of things like krill, because krill are very far down on the food chain, and so they have more energy to offer such a big animal. So here we can see that's like a basic drawing of what's going on. The sun is providing energy for the producers, and producers like plants that are able to utilize that photosynthetic energy, and heat is lost. By the time we get to consumers, we're losing more heat, but that chemical energy is broken down into the next thing. So energy is constantly traveling in one direction. The chemicals in our body are getting cycled, so by the time we get to a decomposer, that can get recycled back and broken down so the plants can use it. So energy travels in one direction and often is lost as heat. Chemicals and nutrients are looped in a cycle, just like similar to the water cycle. Now, all living organisms require something called homeostasis. This is the maintaining of our body in a condition that doesn't like to change. Plants and animals experience this. This means that we are able to live within a certain kind of framework, and anytime our body goes out of that framework, it's not really ideal. A temperature is a great way to think about this. When our body is in homeostasis, our temperature is 98.6 degrees. But when our body goes out of homeostasis, then that means like we have 100-degree fever. Our body can do okay out of homeostasis for a little bit, but when the environment gets a little too extreme, that's when we start to have problems. For instance, if we have a temperature of like 105, this can cause a serious issue. Our body is way out of homeostasis, and many of the proteins and enzymes in our body can only function within that narrow range. And because we're outside of that range, a lot of the proteins and enzymes are going to start to break down, which is something we call denature. If they start to break down, then that means that our body is going to the organs and things are going to shut down. So this is something that we really don't want to see happen. And that's why the first priority of a doctor when they see a patient with like 105-degree temperature is to get their temperature down as quickly as possible. Sometimes they even put them in ice baths to cool them down. But living organisms interact with their environment and respond to changes in the environment. This is another thing that living things are able to do. They have to be able to adapt to their surroundings. All living organisms also must reproduce. The ability to reproduce and have offspring classify something as living. Now, sometimes it can be more complex, like sexual reproduction that requires two organisms. Sometimes it can be very simple, such as asexual reproduction that only requires the one organism to make a copy of itself. But it's still a type of reproduction. Genes are necessary as well for something to be alive. They have to have some kind of genetic information in the form of DNA that is passed on to their offspring. Now, all living organisms have DNA, which is vital to inheritance of material from one organism to the next. And DNA stands for deoxyribose nucleic acid, which we'll get into a lot more in later lessons. All right, so an adaptation, any kind of modification that makes an organism better able to function in a particular environment. So everything that is alive has to be able to adapt. We have to be able to adapt to different situations. Going back to school, if you didn't take classes during the summer, you're adapting back to a different schedule. Adaptation is something that all living things do. And evolution is the change in an organism over time. So eventually, if you have enough adaptations, the organism will be able to create something entirely new, and this is called evolution. So here's an example of penguins who've been able to evolve over time. The penguins used to be able to fly and have wings that suggest that they were once capable of flight, but that is no longer the case. But because they believe in their past that penguins used to be able to fly, when they're swimming through the water, their wings actually make the same motions as if they were flying through the air. So their wings make the same kind of rotational pattern as a bird in flight. And they have the same structure as other birds, even though they no longer fly. But other adaptations are very closely compacted feathers to keep them nice and warm. Extra insulation, and extra flap of skin on their stomach in order to help protect the egg. So the theory of evolution explains the diversity and the unity of life. It suggests that all living things had a common ancestor, and from that common ancestor, things have changed and branched off due to genetic mutations. It's important to remember that genetic mutations are involved. It isn't just something like, oh, you wanted to get exercise, and so you exercised and built up your muscles, and then you had a baby, and the baby had extra big muscles. That's not how it works. It's not built into the baby's genetics. It's something that you have changed physically about yourself. Think of it as also like a tattoo is a good way to describe it. You get a tattoo, your baby's not going to come out with a bunch of tattoos because their genetic structure was not changed. Now, if your baby has a genetic mutation that allows for it to have stronger muscles and be overly strong, then that's different. But it really is about the genetics that's important. So common descent with modification is that throughout time there have been changes, and these changes have led to the creation of new organisms. So natural selection is the evolutionary mechanism proposed by Charles Darwin, and this just suggests that some organisms are better able to adapt to an environment than others. So for instance, there are two moths, and one of them is green and one of them is brown. If the brown one lands on a tree trunk, then it is going to camouflage better, and the green one is going to stand out more. So birds may eat the green one more until naturally there are just more brown ones in the population because they were able to hide better. This allows for the brown one to pass on the brown genes, and eventually the green genes may disappear entirely from the population. So this idea of natural selection by Darwin was proposed based on his review of finches and their beaks in the Galapagos Islands. And the beaks were shaped to eat different things because animals had to adapt in order to survive in different habitats on the Galapagos Islands. The whole idea again behind this is that mutation fuels natural selection. Mutations occur naturally in any environment, in any living thing, and these mutations are going to allow for an organism to change over time. So here's another example, this one looking at a deer eating some plants. In this case, it has a fuzzy plant and it has smooth plants. Well, the deer doesn't necessarily like the fuzzy plant and prefers to eat the smooth plant. So the fuzzy plant is able to pass its genes on to the next generation. Over time, there's going to be more fuzzy plants and less smooth plants because the smooth ones just keep getting eaten and they're not able to reproduce. So the fuzzy ones are changing over time. Even though this is technically still the same species, the fuzzy and the smooth, this one has a gene that is more advantageous to allow it to survive to the next generation. An evolutionary tree is like a family tree, so you start off here with a common ancestor, and anything toward the trunk of the tree is more primitive, and anything out here toward the branches and the tips of the tree are much more advanced. So this is a more primitive common ancestor, and we can see that we have bacteria and what's called archaeobacteria that branched off from that. Archaeobacteria is like bacteria that live in extreme environments like really salty conditions, really hot conditions, things like that. So bacteria are the ones we think of most of the time when we think of bacteria, and they're very primitive organisms that have been able to adapt over time. If you think of staph infections and how staph infections have gotten more and more severe, that's because the bacteria have also been able to change and adapt to antibiotics that we have been giving. Our antibiotics haven't changed in as many years, so that's why we're having antibiotic-resistant strains of bacteria, because they are adapting and changing. Then we have the more advanced organisms, such as the animals, which are one of the more evolved creatures, and you can see how they branched off over time. Here are plants back here and protists and so on. So a common ancestor comes first, and that leads to the branching of two things, in this case bacteria and archaeobacteria. And then you have something else that branches off of this, which is in this case a cell that has a nucleus or a membrane surrounding its nucleus. From there you've got the protist, but that branches off into more complex things like plants, fungi, and animals. This over time is going to cause more changes, and animals and plants are still adapting to this day. It just takes thousands and thousands of years in order for us to see the creation of a new species. So taxonomy is this labeling of the different organisms, and this is an area of study. And systematics is the study of the evolutionary relationship between these organisms. So it's kind of, taxonomy and systematics is kind of like the same thing, but it's just really ways to study how these organisms are related. They can be grouped into categories, and this is a listing of the categories. Species is the most specific, and domain is the most broad. So there are 3 domains, and then there are millions of species, considering both plants and animals. So it goes species, genus, family, order, class, phylum, kingdom, supergroup, and domain. Now, oftentimes you may not hear of supergroup, and also there's things called subspecies. That's because scientists have a lot of arguments about, well, how much classification is too much? Should we even consider something that's a subspecies when technically it's able to reproduce on its own? So there is a lot of debate with classification, even with amongst the animals and plants that are found in classification. So here we can see as well domain, supergroup, and this is saying that supergroups are only present in the domain Eukarya, and some people don't even recognize the use of supergroups. And then a species. Usually the scientific name of an organism has a 2-part name. It's called binomial nomenclature. It consists of the genus and the species. The genus is always capitalized, the species is lowercase, and it's always put in italics if you are typing it out, and if you are writing it out like on paper, then it is underlined. So here we've got the classification for humans, and here's a classification example for corn. We're both in the group Eukarya, which means we have a membrane surrounding our nucleus. But that's where things get different. We're in different kingdoms, and so humans are kingdom Animalia, phylum Chordata, then class Mammalia, primates, Hominidae, Homo, and sapiens. So we are Homo sapiens. And the correct way to write our scientific name, you can abbreviate it H. sapiens, but you can also say Homo sapiens with the genus capitalized, the species lowercase, and everything in italics because it is typed on a computer. All right, so the 3 types of domains, we have domain Archaea, domain Bacteria, and domain Eukarya. Domain Archaea are prokaryotic, and domain Bacteria are also prokaryotic. Prokaryotic again means no membrane surrounding the nucleus. Eukaryotic means they do have a membrane surrounding the nucleus, and this consists of more of the complex organisms that we are most familiar with. But domain Archaea means we've got the more complex, well, they're not necessarily complex, but they're just different types of bacteria. These are the bacteria that are going to be found in groups such as like really extreme environments, really hot conditions, really salty conditions, and domain Bacteria is the more ones that we are commonly familiar with. Domain Archaea, they're prokaryotic, and they are adapted to extreme environment, absorb or chemosynthesize food. So if there is a hydrothermal vent or like an underwater volcano in the bottom of the ocean, then that's where you're going to find these organisms. They do not photosynthesize. They take the chemicals that's in the water and they turn the chemicals into energy. So it's something called chemosynthesis. It's not super common, but chemosynthesis is an alternative form of photosynthesis, just using chemicals instead of sunlight to make energy. Domain Bacteria is the one we are more familiar with. If you have strep, if you have an ear infection, if you have a staph infection, all of that stuff is caused by bacteria. But there's many good bacteria as well. If you take a probiotic for your stomach, that's replenishing good bacteria that lives in our digestive tract. But these are also prokaryotic. Getting into the eukaryotic, we can see the more advanced organisms, the kingdom Protista. These are typically like little kinds of tiny creatures that often live in water. So in pond water, you've got algae, protozoans, slime molds, water molds. You've also got things like Euglena and Paramecium and amoebas. Those are all in Kingdom Protista. Kingdom fungi, mushrooms, and molds and fungus and those kinds of things. Eukarya plants, Kingdom Plantae, and Kingdom Animalia. Those are pretty obvious what they are. So how they're divided up, domain Archaea consists of the kingdom, they're still kind of being determined, but it's usually kingdom Archaea bacteria, and domain bacteria consists of kingdom bacteria, domain Eukarya, you have kingdom Protista, kingdom fungi, Plantae, and Animalia. Supergroups is something that is still not widely accepted, but accepted enough. You've got the more complex Excavata, Chromovelata, Rhizaria, Archaea plastida, Amoebozoa, and Opithconta. Still fun to pronounce those. And this is just kind of an example of different organisms that live in this, algae and slime molds and fungi. It all gets kind of complicated because of the diversity of animals and plants that are in the group Eukaryotics. We already kind of mentioned this, that scientific names are very important because of the binomial nomenclature. Scientific names are universal. So Homo sapiens, if you tell a scientist in the United States versus a scientist in Australia, ask them what a Homo sapien is, they'll automatically know. Most will know Homo sapien. But the idea is that having a common scientific name unifies the living organisms and makes it less confusing and easier to group them. The scientific names are written in Latin. Latin is a language that hasn't changed very much. This is what exactly we want for classification of organisms. Again, the name is binomial, so that means bi meaning two, that there are two parts to the name. The first is the genus, the second is the species. The first is always capitalized, the genus is always capitalized, the species is written in lowercase, and of course it is always italicized if it's written on a computer and underlined if it is written in paper. All right, so moving on to the scientific method. The scientific method is a way that we can explore the environment and a systematic way of doing things so that we can test experiments. Observation, hypothesis, predictions, data collection, and results. Now, depending on what you research or what you think or who you ask, there could be more of a breakdown than this. Like you start with a question, that question or the observation, and then the observation leads to a question, the question leads to a hypothesis, and so it can be broken down into more steps, but these are the basic steps. You're always going to see something in the environment and wonder, oh, I wonder why that's happening, and that's going to lead to the scientific process. This is just showing kind of a flow diagram of how the scientific method works. Conducting the experiment is a vital part of this, and making sure the experiment is conducted in an appropriate fashion. So a hypothesis is an tentative explanation, and it's usually written as an if-then statement. If this happens, then this happens. It's a testable prediction. You want something that can be tested and at the end say our hypothesis was supported or not supported. And you can never say that our hypothesis was proven or not proven because that's not how science works. Very rarely in science does something absolutely true. There's always going to be exceptions to the rule. So a hypothesis is either supported or not supported based on the research that you conducted. So observation, hypothesis, and then something that is testable by using an experiment. The experiment is where all the action really happens, and you can have deductive reasoning within the experiment, and this makes a prediction about the outcome. So you are thinking, I think this is going to happen, but I'm not sure, but this is why I think this is going to happen. And then you perform the experiment to see whether you were correct or not. So experimental design needs to be very detailed and followed step by step in order to be accurate, because the whole idea around this is that you want other people to conduct your experiment and see if they get the same results. Really, you're going to be able to determine if something is more accurate or not the more times the experiment is performed and the more time the results are the same. So experimental design and being as accurate as possible is very important. You have an independent variable. The independent variable is something I, the scientist, am changing. So let's say we were conducting experiments about why a plant is able to bend toward the sun. And we put one plant in the sunlight, one plant just left on the table, and one plant in a closet. The one in the sunlight is actually placed next to a window. And so one in a closet is in complete dark. You want to keep everything the same, but the independent variable that I am changing is the location of the plant. One in the window, one just left on a table, and then one in a dark place. So you really just want to change one thing. You don't want to change the location of the plant and what type of plant you're growing and when they were planted and the soil and the size of the pot. You're changing too many things in that case. You want to keep everything the same except one thing, and that way you know for sure that if there is something changing in the experiment, it has to be due to that independent variable. A test group is the group that you are testing, and that is often known as the dependent variable because the dependent variable depends on the independent variable. You also will always have a control group. The control group is very important. It is what you are controlling. The control group is what you're using to compare your results to. So in the case of our plants that we were talking about, it would be the plant that's just left on the table. It is what you're using as a comparison to what you're comparing the independent groups, the independent variables to. Data is the result that can be collected. It can be in the form of numbers. The plant grew so many centimeters. It could also be in the form of like colors. The plant was yellow versus the plant was green. The plant had flowers. The plant didn't grow any flowers. So you can have different kinds of data. And the more data that you collect, the better, because it means that you have more information to draw your conclusion from. Statistical data is measuring the results and how the results happened. And a standard error is how far off of the average something is. If you have really high standard errors, that means there might be something wrong with your experiment and it might not be super accurate. And the results may not be something you can really depend on. So having a short standard error is better because that means that your data is more in agreement with each other. Statistical probability and statistical significance, this is something that's really important to also understand. Statistical significance means that less than 5% is acceptable. So the probability value is less than 5%. That means that if there is less than 5% of variation, your results can be considered fairly accurate. If there is a lot of variation, we say that there's not statistical significance, and therefore that there is too much range of difference for something to be accepted as supporting the hypothesis. The lower the p-value, the greater the confidence in the results. So the lower the change, the more like, hey, something's really going on here because I didn't notice a lot of differences, a lot of changes. So we've got to look at that compared to the control, and we've got to say, hey, with the lower the p-value, the higher the chance that we can say something's going on. And this is not due to chance alone. So there's always something that could be a chance or a variation in an experiment, but not due to chance alone means that something is going on in this experiment, something significant is happening, and so we can accept that as a change is taking place. Here we can see kind of a basic graph, and here's our standard error bars. When we have this is the average overall, but this is like our highest outlier and our lowest outlier. We don't want a lot of variation. If we have a standard error bar that's all the way out here, that could be skewing our results. We like to have these tiny error bars because that means that everything was closer to the average and our results are more accurate. If we have things that are way off, then that could be throwing the results off, and our results could not be accurate maybe, and that could be due to something that malfunctioned during the experiment. It's really important when doing an experiment that you have a conclusion in peer review. The conclusion is whether you support or do not support your hypothesis based on the results that you see. And you want to always go back to the hypothesis. Our research and data showed that our hypothesis was supported or our hypothesis was not supported. And again, we don't say whether it was right or whether it was wrong, because this is one experiment. This experiment needs to be conducted many, many, many times in order for us to say that there's something right or something wrong. And even then, it's very rare for scientists to do that. I mean, we've got lots of theories out there, like the theory that the Earth is round. And we still don't say that, oh, it's a lie, it's a theory. And there's a theory that the Earth moves around the sun, and we still can't say that, oh, this is how it happens. We can only say that it's a theory. So it's very rare that we say that something happens this way and there is no way that it's not true, because there's usually almost always an exception to the rule. Then, supporting that, you have to get your information out there and present your information to the world, because first of all, somebody could claim your information as theirs and say, hey, I discovered this first, and that's not something you want to do and something you want to happen. So you want to have your information published out there for others to review, also so that others can conduct an experiment similar to yours to see if they get the same results. Sharing information is vital in the scientific world. So you can claim the research as your own, but so that others can try it, or it gives another person an idea and they can branch off and do something else similar, and more discoveries can be made. Scientific publications are a lot different than just some random online article. There are a lot of good, legitimate scientific publications that are now published online. Audubon Society and Evolution Magazine are two examples, but a peer-reviewed article means that it has been reviewed by other scientists, and it's not just some piecemealed together experiment that didn't have good control or follow the scientific method. It is well thought out, well-focused, legitimate experiment. So the difference between scientific theory and scientific law, this is something that also causes a bit of confusion. With scientific theory, concepts that are joined together two or more well-supported and related hypotheses. So this is something that science thinks is true based on a lot of evidence, supported by a broad range of observations, experiment, and data. Lots of things point to this being true. The theory of evolution is a good example. Lots of things point to evolution being true because we can see fossil records, we can see embryological development, we can see this on a smaller scale with the deer eating the leaves like we talked about before. So that's what a theory is. Now, a scientific principle or law is a widely accepted set of theories and no serious challenges to its validity. Oftentimes, a scientific principle or law can be supported by a mathematical equation. And so these are a little bit more valid, a little bit more able to be proven. One way to remember it is a law is what happens and a theory is why it happens. So a law is what happens and a theory is why it happens. So here are some examples of theory. A cell theory is that all organisms are composed of cells and new cells come from only pre-existing cells. So our cells start as one cell and they make a copy of themselves, and so on. And evolution is a theory as well. All living organisms have a common ancestor, but each is adapted to a particular way of life. Examples of controlled studies. So you can see we have a population that gets divided. You perform the experiment. The data is collected, and then you're eventually able to get the information that you need. That information, you go back to the hypothesis. What was our original hypothesis, and was it supported or not supported? So here we can see the hypothesis based on this diagram before is that a newly discovered antibiotic is better treatment for ulcers than antibiotic A, which is currently used. One, you divide the group randomly. Random is very important in science. Also not to be biased. That's another important concept of an experiment, is you don't get to pick which one you like. One control group includes subjects with ulcers who are not treated with either antibiotic, so that's the one that you're comparing both results to. And then you got antibiotic A or an antibiotic B. Perform an endoscopy to discover what the inside looks like, and then they're able to determine whether the ulcer was healed or not, depending on the antibiotic that was used, and they compare that to their group, to their control group. So in general, science is a systematic way of acquiring knowledge about the natural world. And scientists are people who like to have things in an organized pattern. Technology is really helpful with this, and because of technology, we've been able to do a lot more things with science. But it's also caused complications too. We may find a new medicine and may later find out that there is a complication with that medicine that we didn't realize at first. We thought it was safe, and now we realize it's not. So discovering new things is good, but sometimes it can make things worse in the long run. Examples of new technology that can help is cell phones, new drugs, the ability to have internet in more places, the ability to have more advanced like incubators and medicines for helping like little infants and more advanced procedures. All of this is thanks to technology that's available. Climate change can be an issue too. It refers to the change in the normal cycles of the Earth's climate attributed to human activities due to imbalance in chemical cycling of carbon. This is the main thing. We are burning fossil fuels at an enormous rate. Fossil fuels are living organisms from the past that have been compressed over heat and pressure over time. And so the organisms have carbon present, and that carbon is released into the environment. This carbon is a problem for the environment because it causes like a blanket to form over the earth, and it makes it so that the greenhouse effect is experienced and heat gets trapped closer to the surface. Summers are getting hotter, winters are getting warmer, and it's making it difficult for animals to adapt. So the amount of carbon dioxide is making a blanket that heats the earth up by the greenhouse effect. Biodiversity is the total number of living things. Biodiversity is good. Bio for life. Diversity means lots of different kinds. You want lots of different kinds. The more variety there is, the healthier the environment is overall. But we're seeing an extinction take place at an alarming rate, and organisms are starting to die off, with the main cause of extinction being habitat loss. As humans are evolving, we are taking over more and more places, and that means that the animals are losing those places because they're not able to adapt fast enough to survive as their habitats are being lost. Estimated to be losing hundreds of species every year due to human activities. So some other examples: tropical rainforest and coral reefs are home to many organisms, but both of them are being destroyed by human activities. The canopy in the rainforest supports orchids, insects, and monkeys, among other things, and coral reef provide habitats for jellyfish, sponges, crabs, lobsters, and fishes. But as the temperature rises in the oceans, corals are bleaching, and it's causing coral reefs to die. And as humans are expanding into the rainforest territory, we're cutting more and more rainforests down, and that's causing that ecosystem to be threatened as well. But humans depend on a healthy ecosystem. We need it for food, medicines, and just raw materials. So as these places are getting destroyed, we are using up those resources. Draining the wetlands of the Mississippi and Ohio rivers, this actually causes worse flooding damage, ruined farmlands, and destruction of the rainforest is causing more species, and it's decreasing the amount of lumber that's also available. New diseases are also being emerging over the past because we're going into areas that we haven't been before, because we are doing things we haven't been before. Obviously, COVID is a great example of that. Eating things and being near things that we aren't supposed to be can cause for new diseases to develop, and also diseases are changing over time as well. They're adapting to their environment. Diseases come from increased exposure to insects and animals that are trying to survive with less habitat. changes in behavior, uses of technology, globalization, and things changing and mutating on their own. All right, so we finished just in time. And if you have any questions, by all means, please reach out in class. And this is chapter 1, and we will begin chapter 2 next time. Thank you for your attention.